On the behalf of Student Scientific Association of the Medical University of Silesia, in cooperation with Doctoral Student Self-Government of the Medical University of Silesia and the Polish Dental Student Association, it is a great honor and pleasure to greet you all at the closing ceremony of the 69th edition of the International Medical Congress of Silesia, SIMC 2023. We thank you very much, authorities of our university, for coming. The prorektor for prorektor do spraw nauki i współpracy międzynarodowej, profesor doktor habilitowana nauk medycznych Katarzyna Mizia-Stec. Prorektor do spraw studiów i studentów, profesor doktor habilitowany nauk medycznych Jerzy Stojko. Dziekan Wydziału Nauk Medycznych w Katowicach, profesor doktor habilitowany nauk medycznych Tomasz Francuz. Prodziekan do spraw studenckich Wydziału Nauk Medycznych w Zabrzu, pan doktor habilitowany nauk medycznych Andrzej Tomasik. We also greet the curator of the Student Scientific Association, profesor doktor habilitowany nauk medycznych Michał Cholecki. And our honorary guest, Dr. Ali Javaid, MD, PhD. Thank you very much for honoring us with your presence. We also want to thank you, all the members of the scientific committees, for joining individual sessions and spending your time with us. We greet all present, active and passive participants of our Congress. It is a great pleasure for us to receive abstract on such high scientific level from many societies from universities in Poland and abroad. We also thank our patrons, sponsors and partners. Our honorary auspicious of the Rector of the Medical University of Silesia, Professor Tomasz Szczepański, the Honorary Auspicious of the Ministry of Health of the Republic of Poland, Adam Niedzielski, Honorary Auspicious of Ministry of Education and Science of the Republic of Poland, Dr habilitowany Przemysław Czarnek, the Honorary Auspicious of Marshal of Silesian Voivodeship Jakub Haustowski, the Honorary Auspicious of Mayor of Katowice City, Dr Marcin Krupa, the Honorary Auspicious of Silesian Pharmaceutical Chamber, the Honorary Auspicious of Polish Young Academy of Polish Academy of Sciences, the Polish Chamber of Physiotherapists. We also thank our media patronage of Forum Akademickie, Dietetycy.org.pl, TVP3 Katowice, TVS, biologhelp.pl, biotechnologia.pl, Dziennik Zachodni, Nasze Miasto and e-Doktorant. Then we also thank our sponsors of the Congress, without whom this Congress we cannot do this Congress. We thank Górnośląska Fundacja Onkologiczna, MedCases, Lepolek, Medycyna Praktyczna, Edra Urban & Partner, Więcej niż Lek, MedCoach, Medu, Farmi Campus Mind and Physio. And also our scientific patronage, Polskie Towarzystwo Radioterapii Onkologicznej, Societas Anatomica Polonium, Polskie Towarzystwo Medycyny Ratunkowej, Polski Instytut Evidence-Based Medicine, Polskie Towarzystwo Alergologiczne, Polskie Lekarskie Towarzystwo Radiologiczne, Polskie Towarzystwo Nadciśnienia Tętniczego, Fizjoterapia Polska, Polskie Stowarzyszenie Implantologiczne, Klub Młodych Nefrologów, Polskie Towarzystwo Nefrologiczne, Polskie Towarzystwo Neurologów, Polskie Towarzystwo Onkologiczne Oddział Śląsk, Polskie Towarzystwo Diabetologiczne Oddział Śląsk, Oddział Śląski Polskiego Towarzystwa Fizjologii Klinicznej, Śląski Oddział Towarzystwa Onkologii Klinicznej, Oddział Śląski Polskiego Towarzystwa Ortopedycznego i Traumatologicznego, Polskie Towarzystwo Zdrowia Publicznego Oddział Śląsk, Polskie Towarzystwo Neurochirurgów Oddział Śląsk. And also our partners, our partner congresses, International Students Scientific Congress, Juvenes Pro Medicina, International Students Congress, Warsaw International Medical Congress, Oporto Biomedical Summit, Bukowinian International Medical Congress, International Students Conference Riga Stradins University, AIMS Meeting, International Congress of Young Medical Scientists, and Sky meeting. And also, we very thank the Ministry of Education and Science for financing this Congress from the program Doskonała Nauka. Now, please the Rector, Vice Rector for Science Affairs for summing up the Congress.
Dear students, dear professor, dear authorities, dear guests, it's for me a great pleasure, first of all, to be here. This is the unusual evening for our university. This is Students' Scientific Feast in Medical University of Silesia. On behalf of the rector of our university, Professor Tomasz Szczepański, I would like to cordially congratulate to all participants and organizers of this Congress. I'm sure that all you are winners, regardless of the final decision of jury, of distinctions and some others diploma. I'm sure that it is a great evening, it was a great meeting. And I'm sure because I have a long-term experience in these congresses. I have been for more than 10 years the curator of your scientific society, and it was and it is for me a great pleasure to be here. I would like to congratulate all participants, especially from other universities, and I hope it is your first but not the last stay at our university. And last but not least, great congratulations for all organizers, extraordinary students, and IT team of our university. It was a great job. I wish you all a great evening. Thank you very much.
Thank you very much. While organizing the International Medical Congress of Silesia, we have met many challenges. But with the help of many people, especially our university authorities, administration, employees, and volunteers, we have created this Congress again. The novel thing was that during the 69th edition of SIMC, we have managed to organize 11 workshops of the topic of writing scientific paper, discovering molecular gastronomy, going deeper with laparoscopy, elderly simulators, hand masks, body composition analysis, versatile physiotherapy, surgical suturing, unique experience at Zoom virtual clinic, your phone microbiome, and ultrasonography. Thank very much for all partners who help us with organizing those workshops. As each year, the Student Scientific Association has admitted the Franciszek Kokoc Award for the academic teacher who has rendered outstanding service to the Student Scientific Society with their activities, who exemplarily perform their duties as academic teachers, NJ authorities, students, and colleagues, excel in carrying out the task undertaken with the framework of the activities of the Student Scientific Society, and thus making an outstanding contribution to the development and promotion of student scientific activities. Now, please, the curator, to give the to help us giving the award. The winner of this year's award is Mr. Doctor of Medical Sciences, Professor Sum Justyna Poprocka, head of chair of the clinic and head of the chair and clinic of the chair of child neurology in Faculty of the Medical Sciences in Katowice. Congratulations. Thank you very much. And now is time for a curator lecture. Every year during a SIM, a guest speaker is invited to give a lecture. This year we have a pleasure to hear 2 lectures. The first is the curator lecture by Professor Michał Cholecki, superintendent of the STN, with the title Kidney and Eye, Two Brothers.
Szanowni Państwo, bardzo dziękuję za możliwość wygłoszenia tego wykładu honorowego. To jest zawsze dla mnie olbrzymia przyjemność, żeby się z Państwem spotkać na koniec i to jest dla mnie też bardzo ważne. Drodzy Państwo, nerka, oko, 2 bratanki. Gwoli ścisłości. Jak gdyby ktoś nie wiedział, to nerki są narządem parzystym. Zwykle przypominają ziarno fasoli, ale oczywiście ten kształt jest osobniczo zmienny. Na przykład u koni prawa nerka ma kształt sercowaty, u bydła niektóre nerki mogą mieć nieregularnego owalu. A gdybyście siebie zapytali, skąd się wzięła nazwa nerka, to jak widzicie zostało to zapoczątkowane z języka wysoko-staroniemieckiego nirka, czyli nire, nerka. Potem została przyswojona przez przedrostek K i od roku mniej więcej 1500 mówimy nerka. Drodzy Państwo, od górnego bieguna prawej nerki do lewego oka jest 56 cm, do prawego oka 51. Oczywiście nie ma to żadnego znaczenia, ale chciałem skupić Państwa uwagę. Pierwszą osobą, która tak naprawdę zajęła się połączeniami między nerką a okiem, był Richard Bright. To był taki angielski lekarz, patofizjolog, który po raz pierwszy zaobserwował pewnego rodzaju koincydencję, że u chorych na przewlekłą chorobę nerek postępowała ślepota. A drodzy Państwo, dlaczego taki temat? Dlaczego nerka, oko, 2 bratanki? Dlatego, że zarówno nerki, siatkówka rozwijają się zwykle na tym samym etapie embriogenezy. Co więcej, podążają tymi samymi szlakami rozwojowymi. Żeby tego było mało, to ta bariera przeznaczenia kłębuszkowego oraz to połączenie siatkówkowo-naczyniówkowe są strukturalnie bardzo podobne. Tak naprawdę, gdybyśmy nie znali anatomii, powiedzielibyśmy, że jesteśmy w tym samym miejscu, więc albo w nerce, albo w oku. Co więcej, i kłębuszki nerkowe, i warstwa ta naczyniówkowo-siatkówkowa są wypełnione włóśniczkami, czyli znowu pole manewru dla krążenia. I zarówno ten podocyt, który jest czynnościową komórką nabłonka nerek, i komórki nabłonka barwnikowego siatkówki zależą od działania rzęsek na ich powierzchni. Zobaczcie Państwo, ile jest połączeń tak naprawdę albo podobieństw i oka, i nerki. I teraz zobaczcie, jeżeli mówimy o kapsuły, tu jest torebka łamana, tu jest rogówka, tak? Czyli jest kapsułka. Pęczki naczyniowe. Te same receptory, takie samo prężność parcjalna tlenu. Potem mamy błonę. Tu oczywiście mamy połączenie między rdzeniem nerki, a tutaj mamy błonę brucha. I znowu pęczki naczyniowe, podobne cienie parcjalne, podobne receptory. Olbrzymie podobieństwo. Przekrój kłębuszka nerkowego. Macie Państwo naczynie, są fenestracje, są wyrostki stopowate podocytów, jest wreszcie podocyt, jest błona podstawna. Mamy siatkówkę. Są komórki siatkówki, które przypominają podocyt. Tutaj są wyrostki stopowate, tutaj są rzęski. Jest błona podstawna. Są fenestracje. Jest naczynie krwionośne, Bardzo duże podobieństwo. Przy odrobinie wyobraźni, patrząc na dno oka, można sobie wyobrazić, że tak również może wyglądać kłębuk nerkowy. Oczywiście każdy ma różną wyobraźnię, ale to można zauważyć. Cały dowcip polega na tym, że to połączenie między nerką a okiem jest bardzo istotne. Są podobne czynniki ryzyka, podobne patomechanizmy prowadzące do różnych schorzeń. I zobaczcie Państwo, gdybyśmy wzięli pod uwagę na przykład czynniki ryzyka dla przewlekłej choroby nerek. Wiek, palenie tytoniu, obecność cukrzycy, nadciśnienia, otyłości czy hiperlipidemii, tak samo przekłada się na wystąpienie jaskry, na wystąpienie zaćmy, na wystąpienie zwyrodnienia plamki. Podobne mechanizmy prowadzą do obydwu schorzeń. Co dalej? Miażdżyca, uszkodzenie śródbłąka, stres oksydacyjny, stan zapalny, uszkodzenie układu Rea. To samo patomechanizmy w przypadku różnych schorzeń gałki ocznej. Podobieństwo jest. I zobaczcie Państwo, że te wszystkie połączenia są samozbieżne. Czy mówimy o genetyce, czy o kardiometabolicznych czynnikach ryzyka, czy o szeroko pojętym stresie oksydacyjnym. Jeżeli będziecie Państwo możliwe patomechanizmy łączące przewlekłą chorobę nerki, na przykład zwyrodnienie plamki, to zobaczcie Państwo, że wczesne MD występuje 3-krotnie częściej u chorych, którzy mają upośledzoną czynność nerek. Im ona jest gorsza, tym to postępuje szybciej. Gdyż weźmiecie Państwo pod uwagę przewlekłą chorobę nerek i retinopatię cukrzycową, to oczywiście wszystkie zmiany jak mikrotętniaki, wysięgi, kłębki waty występują znacznie częściej u chorych na przewlekłą chorobę nerek i są niezależne od innych czynników ryzyka zmian ocznych. A retinopatia z kolei powoduje zwiększa ryzyko wystąpienia przewlekłej choroby nerek, bo jedna i druga opiera się o tak zwaną mikroangiopatię. Jeżeli chodzi o jaskrę, to u chorych na przewlekłą chorobę nerek występuje zwykle większe ciśnienie śródgałkowe i tak samo częściej obserwuje się jaskrę u chorych na przewlekłą chorobę nerek. Jeżeli chodzi o zaćmę, to co prawda te badania nie pokazują istotnych powiązań, ale tu są zdania podzielone. Część naukowców twierdzi, że zaćma częściej występuje u chorych na przewlekłą chorobę nerek, co wiąże się na przykład z zaburzeniami gospodarki wapniowo-fosforanowej. To są akurat przykłady AMD, retinopatii czy jaskry u chorych na przewlekłą chorobę nerek. Ale istnieje takie pojęcie jak zespoły oczno-nerkowe, które łączą znowu nerkę z okiem. To są wrodzone różnego rodzaju schorzenia nerek, które można zobaczyć patrząc choremu głęboko w oczy. Bo na przykład zespół Wager. Zespół Wager to jest guz Wilmsa. To jest guz Wilmsa z nowotworem występującym przede wszystkim u dzieci, który na całe szczęście dobrze rokuje, ale też jest beztęczówkowość, są anomalie narządów płciowych, jest niepełnosprawność intelektualna i co ciekawe, zagrożenie jaskrą takiego dziecka jest bardzo duże i co najgorsze, że ta jaskra w większości przypadków ma postać ciężką. I zespół von Hippel-Lindau, kolejna sytuacja, która ma powiązania z mutacjami, to są mnogie nowotwory. Czy to mówimy o mózgu, rdzeniu kręgowym, siatkówce czy rdzeniu nadnerczy i różnego rodzaju patologie również obserwowane w nerkach, chociażby rak jasnokomórkowy nerki. Podobny mechanizm i podobne połączenie oka z nerką. To na przykład są drobne tętniaki, szczególnie na przykład w zespole Bourneville-Pringla, który również te dwa schorzenia łączy, czyli stwardnienie guzowate. To są pacjentki, które mają triadę objawów. Najczęściej jest to upośledzenie intelektualne. To jest w różnym stopniu się predysponuje. Padaczka czy naczynkomięśniaki, guzki hamartomatyczne, siatkówki, to co Państwu pokazałem na poprzednim slajdzie. Natomiast to, co jest najciekawsze, angiomielipoma jest takim naczyniowo-mięśniako-tłuszczakiem. To jest łagodny, zwykły nowotwór nerki, który w przypadku zespołu Bourneville-Pringla występuje po pierwsze ale z dużo większą częstością niż w populacji ogólnej, przekształca się w raka jasnokomórkowego. Więc że u takiej pacjentki, która ma zespół Bourneville-Pringla i ma mnogie angiomielipoma, tak chora często wymaga usunięcia obydwu nerek i najczęściej w tej hispacie potem znajdują się ogniska raka jasnokomórkowego. To jest taki właśnie przykład hamartomy u pacjentki z zespołem Bourneville-Pringla. Zespół renal-koloboma syndrom, czyli obustronny ubytek błony naczyniowej oka. I znowu występuje z hipoplazją nerek albo z wadą, taką jak wstecz otwór pęcherzowo-moczowodowy, który predysponuje do nawracających zakażeń układu moczowego. Zespół Alporta, który łączy nerkę z okiem, dlatego że pacjenci mogą mieć nie tylko zaburzenia słuchu, ale objawy oczne, stożek soczewki, zaćmę, zwichnięcie soczewki. Czyli znowu macie Państwo połączenie nerki z okiem. I tak wygląda mniej więcej stożek rogówki, a tak stożek rogówki wygląda w różnego rodzaju badaniach, czy w kamerze Pencam, czy objawy kropli oleju, czy w lampie szczelinowej. Ale macie Państwo chorych na przewlekłą chorobę nerek. Przewlekła choroba nerek ma to do siebie, że po pierwsze postępuje niezależnie od tego, czy czynnik sprawczy jeszcze jest, czy go nie ma. Po drugie, przewlekła choroba nerek jest jedną z najczęściej występujących chorób, które predysponuje do ryzyka sercowo-naczyniowego. Jeżeli ktoś ma przewlekłą chorobę nerek i ma stadium trzecie, to taki chory ma 10 większe szanse na to, że zginie z powodu udaru i zawału, niż na to, że dożyje leczenia narkozastępczego. Jak ktoś dożywa leczenia narkozastępczego, to i tak jest szczęśliwy, bo większość chorych umiera znacznie wcześniej. A w ciągu najbliższych 12-15 lat przewlekła choroba nerek będzie piątą po chorobach sercowo-naczyniowych i chorobach płuc przyczyną zgonów na całym świecie. Jeżeli chory dojdzie do izoterapii, to ma mocznicę. Jak ma mocznicę, to ma różnego rodzaju reakcje. Ma odwarstwienie nabłonka, obrzęk plamki, krwotoki siatkówkowe, kardiopatię taśmowatą, reakcje zapalne, ból, zaczerwienienie, tak typowe dla chorych z zaawansowaną przewlekłą chorobą nerek. I dzieci często mają kalcyfikację i kalcyfilaksję. Często są zgładzone złogu wapnia, nie tylko w skórze, nie tylko w naczyniach, ale również na przykład w rogówce. To Państwo na tym slajdzie widzicie. I tak dobrze znana retinopatia. Ciekawostka. Jeżeli ktoś ma te pierwsze cukrzycy, to w zasadzie nieobecność retinopatii przemawia za rozpoznaniem niecukrzycowej choroby nerek. U chorych na typ drugi cukrzycy już tak łatwo nie jest, bo to, że chory nie ma retinopatii, wcale nie znaczy, że ma niecukrzycową chorobę nerek. Więc to jedno drugiego niestety nie wyklucza. Dlatego zalecenia Polskiego Towarzystwa Diabetologicznego bardzo skupiają się na cukrzycowej chorobie oczu. Macie Państwo dokładne wytyczne, kiedy co robić, czy w typie pierwszym cukrzycy, w typie drugim cukrzycy, kiedy te oczy kontrolujemy, jakie możliwości leczenia. Jeżeli chory ma, na przykład nie ma retinopatii, co kontrolujemy go co 2 lata. Jeżeli ma proliferacyjną, czyli bardzo ciężką, to wymaga pilnej laseroterapii. Te wszystkie wytyczne są, ale oczy musimy dbać szczególnie o chorych na cukrzycę. I co ciekawe, ta regularna kontrola dna oka u chorych na cukrzycę umożliwia zapobieganie utracie wzroku u większości chorych. Zobaczcie Państwo, 98% chorych może uniknąć utraty wzroku, jeżeli będzie dobrze kontrolowana i ewentualnie dobrze leczona. Moi drodzy, wzrok to jest taki król zmysłów. Wykorzystujemy go przez całe życie w różnych sytuacjach. Powłóczyste spojrzenie z rzęs, szkoła uwodzenia. Ostry, przeżywający wzrok, oczekujemy posłuszeństwa, tak jak przykład ja od moich asystentów na wizycie. Dobry wzrok ułatwia odnajdywanie się w przestrzeni, czuwa nad naszym bezpieczeństwem, nie przewracamy się. Spróbujcie na przykład jeść z zamkniętymi oczami albo udajcie się do restauracji tak zwanej ciemnej, tak, gdzie nie widzicie nic, tylko wyostrzają Wam się zmysły. Jest takie zupełnie proste. I to, czy Wy widzicie, czy nie, to nam ułatwia codzienne funkcjonowanie. Kontrolujemy przebieg, efektywność naszego działania. I tak naprawdę za pomocą wzroku odbieramy większość informacji otaczającego nas świata. Oczywiście możemy kompensować pewne niedobory za pomocą słuchu, za pomocą dotyku, za pomocą smaku. Ale są takie informacje o świecie, których nie jesteście bez wzroku w stanie ogarnąć. Nie możemy dotrzeć poprzez inne zmysły. Nie usłyszycie kolorów, nie dotkniecie ptaka w locie, nie powąchacie widoku zachodzącego słońca. Jako ciekawostkę José Saramago, portugalski pisarz, który otrzymał Nagrodę Nobla, powiedział, że spędził życie patrząc ludziom w oczy, że jest to jedyne miejsce na ciele, w którym być może wciąż istnieje dusza. Dziękuję Państwu za uwagę.
Thank you very much for this lecture. And now time for the thing all of you are waiting, the Scientific Session Awards. Despite the high level of the all work present, only the best can receive a prize. Therefore, without further ado, we begin the award ceremony for the winners of the SIMC 2023 Scientific Session. Can I please the Vice Rectors to help us giving the prizes? Okay, so let's begin. The first session is the session of oncology. Can you open the results? During the awards announcement, please come upstairs on the left side and go down on the right side. Also stay to the end of all awards in each session. We will make a photo of all awarded participants of each session. So let's start with oncology. The second prize goes to Anna Kozub and Agata Suleja for the work postoperative chemotherapy in sigmoid colon and sigmoid rectal fold cancer patients. And now the first place. The first prize goes to Natalia Hajduga and Katarzyna Jaśkiewicz for the work hyperbaric oxygen therapy as a complementary Okay, can we please the decan and vice decanów to help us giving the lectures? The prizes, sorry. And now let's move to the session of orthopedics. The second prize goes to Antoni Straszczak, Paweł Reterski, and Aleksandra Skorupa-Strojna for the work Effect of Restricted Blood Flow on the Bioelectrical Activity of the Quadriceps femoris during squat exercises. And the first prize goes to Łukasz Dylewicz, Jakub Rudzki, Kacper Solarz, Karol Demel, and Jakub Będkowski for the work Analysis of ACL Surgical Treatment and Postoperative Outcome Retrospective Study. Thank you very much. And now let's move to the session of gynecology. The first prize goes to Urszula Guzik, Wiktoria Śliwka, Julia Szostakow, Maria Dziędziel, and Kamila Deptala for the work Knowledge of and Attitude of Polish Society Towards In Vitro Fertilization as a Method of Infertility Treatment. Thank you very much. And the second prize goes to Wiktoria Laskowska and Angelika Burzy for the work Knowledge of Fertility Recovery After Pregnancy Among Women in Poland. Maybe also the first place to speed it up a little bit. And the first prize goes to Joanna Kaźmierczak, Klaudia Hachuła, Iga Szymańska, Karolina Piskorowska, and Marta Kowalska for the work The Knowledge about Nutrition and Supplementation During Pregnancy Among Polish Women. Thank you very much. And now this. And now the session of non-invasive cardiology. The first prize goes to Agnieszka Adamiec, Bartosz Basiaga, Dominika Bieczek, Aleksandra Cader, Adrianna Ściślicka, and Monika Wandasiewicz for the work Preclinical History of Patients with HCM Reflecting the Course of the Diagnostic Process. And the third prize goes to Aleksandra Urbaś, Paulina Wietrzycka, Agnieszka Partyka, Jędrzej Jankowski, and Oliver Piechota for the work Vascular Arrhythmia, Clinical Pathological Characteristics. The second prize goes to Aleksandra Machnik, Julia Dołęga, Mateusz Drabczyk, and Michał Górecki for the work Exercise Loss in Asymptomatic Severe Aortic Stenosis, a Preliminary Study. The first prize goes to Marcin Schulz, Julia Cieśla, Michał Krawiec, and Piotr Kuczera for the work Vascular Stiffness and Endothelial Function in Patients with Coronary Artery Disease and Diabetes. Thank you very much. And now the session for invasive cardiology. The third prize goes to Michał Kapałka and Michał Krawiec for the work Results of Transcatheter Closure of Patient Ductus Arteriosus in Neonates and Infants Less than 6 kilograms. The second prize goes to Georgi Bokeria for the work Aortic Valve Neocuspidization with glutaraldehyde treated autologous pericardium Ozaki procedure in Georgia. And the first prize goes to Kamila Florek and Michał Błaszkiewicz for the work Comparison of two techniques for the treatment of calcified coronary artery. So now let's move to the session of neonatology and pediatrics. The third prize goes to Weronika Rossler, Klaudia Sobik, Klaudia Hachło and Natalia Lewoniuk for the work Characteristics of rotavirus infections in the region of Silesia between June 2019 and August 2022 in terms The second prize goes to cerebellar mutism after resection of midline cerebellar or intraventricular posterior fossa tumors in children by Michał Schroeter, Izabela Rosu, Jakub Rudzki, Marcin Ciekalski and Antonina Radolińska. And the first prize goes to Julia Osińska, Magdalena Macura, Magdalena Nowak and Julianna Schwarzenberg for the work Biological Therapy in Children with IBD Single Center Study. Very much. And now the session of neurology. The third prize goes to Artur Stasiniewicz by the work The Assessment of the Concentration of Pentraxin-free Osteocalcin and Osteopontin in a cerebrospinal fluid in patients with MS. The second prize goes to Dominika Wrubel and Kaja Zdrojewska for the work 1 year follow-up of drip and ship versus mothership transport models of mechanical thrombectomy. And the first prize goes to Marek Misiak by the work Morphological Analysis of Normal Human Lumbar Dorsal Root Ganglion B Ganglia and Where to Find Them. Now let's move to the psychiatry. And the first prize goes to Magdalena Stensel and Błażej Pilarski for the work Does the Weather Affect Suicide Rates? The second prize goes to Zuzanna Mielczarek, Karolina Kamińska and Michał Ciołek for the work An Analysis of Polish Psychiatrist Consultations of Displaced People from Ukraine. The first prize goes to Karolina Kamińska, Michał Ciołek, Izabela Rosu, Magdalena Matlakiewicz and Maria Potaczek by the work Symptoms of Bipolar Disorder Among Adolescents Residing at Youth Education Centers in Silesia in Poland. Thank you very much. And now please the representative of Polish Dental Students Association, Karolina Ziaja, because we are going to the session of dentistry. And the first prize goes to the second, because there wasn't so much work, to Paulina Musiał, Magdalena Błasiak, Małgorzata Imiołczyk, Aleksandra Gorzyp, and David Hochu for the work Dental Age Estimation in Silesian Children with Demirillians Method. And the first place goes to Katarzyna Janik, Antoni Świątkowski, Małgorzata Rymarczyk, Zuzanna Galicka for the work A Multidisciplinary Approach to Enhancing the Welfare of Patients in Maxillofacial Surgery. Okay, now the session of systematic reviews and meta-analyses. Let's move to the third prize for the work entitled The Lizarov Technique, a Review, prepared by Olaf Hajnus, Patrycja Kłaptocz, and Katarzyna Łukoś. The second prize goes to Kamil Kania and Radosław Karaś for the work Arteria lusoria, a marker of fetal abnormalities and the cause of multiple clinical manifestations. And the first prizes, the first prize goes to Anna Kozub, Aleksandra Nasiek, Marta Spruś, and Łukasz Czogalik for the work Surgical Management of Multiple Brain Metastases of the Tumors of Unknown Origin, a systematic review. And the second goes to Marcin Goławski, Piotr Lewandowski, and Iwona Jabłońska for the work The Reassessed Potential of SARS-CoV-2 Attenuation for COVID-19 Vaccine Development, a systematic review update. Pierwszego autora nie ma.
Zapraszamy, Piotrze.
And now, please, Ola Mroskowiak, the president of the doctoral self-government, because we are entering the doctoral session. First, the session of experimental medicine. The first prize goes to Katarzyna Szałapska-Rąbała for the work Honickel and Magnolol, a promising compound in the treatment of diabetes and its complications. The second prize goes to Irina Popova and Denis Stoliar for the work Variant Anatomy of Anterior Neck Region at Prenatal Period of Human Ontogenesis. And the second place goes to Karina Maciak, Angelika Dziedzic, and Joanna Saluk for the work Prothrombotic and Pro-Inflammatory Phenotype of Blood Platelets in Secondary Progressive Multiple Sclerosis. And the first place goes to Jonasz Dragon for the work Behavioral Alteration after Chronic Administration of Escitalopram in the rodents subjected to early life stress. Thank you very much. And now the session of clinical medicine. The third prize goes to Barbara Janota for the work Lifestyle of Women with Hashimoto Disease, Identifying the Areas that Require Changes. The second prize goes to Sabina team for the work Physical Activity and Pelvic Floor Muscle Training in Postpartum Women. And the first prize goes to Przemysław Witek and Patrick Zemła for the work Optimizing of Biological Treatment of Ulcerative Colitis First Step. And now the session on Public Health and Healthcare. The third prize goes to Mateusz Szymała and Adrianna Papierok for the work Noise Assessment in Kindergarten. The second prize goes to Marta Dziędziel, Angelika Gąsiorczyk and Bartłomiej Bielas for the work The Patient's Knowledge about Prevention, Treatment and Monitoring of Blood Pressure Disorders. And the first prize goes to Justyna Danel, Paulina Kukla and Paulina Kumienga for the work Glass Sailing in Academia, Women's Career Paths at the Medical Faculties in Poland. Sorry, it was the second, and the first prize goes to Paulina Rutecka, Dawid Wolak, Zuzanna Zięba, Filip Klimas, Dorota Zatłoka for the work Vaccinological Prophylaxis of Seasonal Diseases among Medical Students, Influenza and COVID-19 Vaccination. Thank you very much. And now the very special session, the first time the high school session at our Congress. The first prize goes to Marlena Smerczek by the work The Influence of NaCl at Different Concentration of the Development of Lemna Minor L. The second prize goes to Michał Żelazik for the work The Relationship Between Sleep, Working Memory and Frequency of Mind Wandering Among High School Students. And the first prize goes to Aleksandra Kobyłecka for the work Potential Use of Apple Cider Vinegar and Grapefruit Extract in the Prevention of S. epidermidis endocarditis. And now the session of systematic review and meta-analysis 2. The third prize goes to Victor Nekhemsinachi Oti, Marian Abubakar, Sopu Puruchi Angel Chidi Ikei, Mari Help Chisom Ezeshifoch for the work Evaluation of the Effectiveness and Safety of Colchicine in Prevention of Atrial Fibrillation. The second prize goes to Joanna Zygadło and Grzegorz Procyk for the work Autoantibodies in Atrial Fibrillation State-of-Art Review. And the first prize goes to Grzegorz Procyk and Przemysław Kasiak for the work Coach COVID-19 Influence on Athletic Cardiopulmonary Health, Systematic Review and Meta-Analysis. Thank you very much. And now the session of Public Health and Healthcare 2. The third prize goes to Gabriela Klimkowicz, Izabela Kapuścińska, Magdalena Żegleń, Aleksandra Rajda, Karolina Łodziana, and Alicja Michalska for the work Evaluation of the Antibacterial Activity of Selected Phenolic Acid Against Staphylococcal Reference Strains. The second prize goes to Sabina Kadłubek and Jakub Kamiński for the work A Comparison of Awareness of Cardiovascular Risk Factors between Medical Students and Students of Other Majors. And the first prize goes to Julia Woźniak, Maria Partyka, and Sabina Kadłubek for the work The Symptoms of Chronic Venous Disease are a serious problem in a population of young women. And now the session of basic science, biotechnology and bioengineering. The second prize goes to Wiktoria Suchy, Katarzyna Chwaleba and Mateusz Giełczyński for the work Enteric Nervous System and Pain Perception in Activity-Based Anorexia Animal Model. And the first prize goes to Alicja Kamińska by the work Analysis of Drug Release from Spheres whose degradation depends on MPM in a 3D model of neoplastic invasion. And now the session of anesthesiology, emergency medicine, and intensive care. The second prize goes to Jan Herzyk and Dawid Wilczek for the work Hematologic System Dysregulation in Sepsis Patients with Anemia, a retrospective cohort study. And the first prize goes to Fabian Wesołek, Robert Latacz, and Wiktoria Staniszewska for the work What Blood Pressure and Vasactivity Agents Tell Us About Capillary Refill Time, a prospective analysis. Thank you very much. And now the session of surgery. The third prize goes to Magdalena Górska and Paweł Paziak for the work Pancreatic Serous Cystic Neoplasm, Clinico-Pathological Characteristics. The second prize goes to Michał Serafin, Paulina Kluszczyk, Aleksandra Tobiasz, Tomasz Kowalczyk, and Sebastian Maślanka for the work Pancreatic Post-Inflammatory Cysts Surgical Treatment. And the first prize goes to, so the second prize goes to Agnieszka Portyka, Paulina Wietrzycka, and Wiktoria Bajek for the work Pancreatic Solid Pseudopapillary Neoplasm, Clinico-Pathological Characteristics. And the first prize goes to Jakub Ciesielka and Karolina Majewska for the work The Results of Surgical Treatment and Long-Term Survival of Patients with Ampullary Adenocarcinoma. Thank you very much. And now the session of internal medicine. The second prize goes to Julia Ślężek, Wiktoria Skowron, Zuzanna Szatkowska, and Błażej Szymczuk for the work Analysis of Clinical Efficiency of Omalizumab, Benralizumab, and Mepolizumab in Asthma Treatment. And the first prize goes to Dawid Wilczek and Jan Herzyk for the work The Appropriateness of Red Blood Cell Transfusion, a retrospective study in large teaching hospital. Thank you very much. And now the session of dietetics and nutrition. The third prize goes to Carolina Leopold for the work From Green to Brown, Antioxidant Potential of Coffee Depending on the Roasting Degree. The second prize goes to Kamil Monkosza for the work Students' Knowledge of Selected Diet-Related Diseases and Their Prevention. And the first prize goes to Julia Lewandowska for the work Plan Adaptogens as Natural Antioxidant and Potential Anti-Aging Agents. And now the session on pharmacy and clinical pharmacology. The second prize goes to Arkadiusz Sokal for the work Analysis of the Molecular Structures of Orthopropagyl Derivatives of Galangin and Campeferol. And the first prize goes to Ines Laurenko and Anna Neves for the work Effects of Tamoxifen Therapy of Female Rats on the Expression of Neurotransmitters in Hippocampal Formation. Let's move to the session of physiotherapy. Third prize goes to Jolanta Płonka and Mateusz Krzempek for the work The Problem of Kinesophobia in Overweight and Obese People. The second prize goes to Barbara Lewicka and Piotr Gomuła for the work Performance in Activities of Daily Living Before and After Rehabilitation in a Group of Elderly Patients. And the first prize goes to Natalia Ślęziak, Anna Stalka, Paweł Ganczewski, and Tomasz Stawiarski for the work Music, Friend or Enemy or Fast Reactions. And the last session of radiology, the first prize the first prize of physiotherapy. The second first prize goes to Urszula Skalska and Jan Stopka for the work The Hidden Cost of Being a Musician, the Assessment of the Frequency and Determinants of Pain. And now the last session of radiology. The second prize goes to Iga Szymańska, Marcin Siekalski, and Jakub Mordarski for the work Retrospective and Statistical Analysis of Hand and Forearm Injuries in the Silesian Pediatric Population, a cross-sectional study of post-traumatic X-rays in 2022. And the first prize goes to Marcin Ciekalski, Izabela Rosu, Michał Schletter, and Antonina Radolińska for the work Challenges and Effectiveness of Mechatronical Thrombectomy in Acute Stroke, a Single Center Study. Thank you very much. It was the last session. Congratulations to all winners and all participants who presented the work. Thank you very much. And now let's move to our honorary lecture. We welcome Dr. Ali Javait from the Nensky Institute in Warsaw with the lecture Epigenetic Basis of Neuropsychiatric Disorders, Many Lives, One Master. Dr. Ali, the stage is yours.
All right. Thank you very much. Thanks for this very kind introduction, Piotr, and thanks for the invitation to SIMC. It's definitely an absolute honor and pleasure to be here. It's my third year in Poland. I had a career which started back in US, and then I moved to Switzerland, eventually ended up in Poland. And what I'll present today sums up, like, the topic which I've been working on for the last 9 years or so. I'll try to make it a little interactive. I know it's not a workshop, it's a lecture, but still, we'll make it slightly interactive. So we start with, has anyone read this book over here? It's a very interesting book. It's actually an anecdote by a psychiatrist, Brian Weiss, who's a very peculiar psychiatrist, Well, at least he became very peculiar. He used to be a very conventional one, but then he changed his thinking. And he describes the case of one of his patients who suffered from inconsolable anxiety. Everything was fine in her life. There were no apparent risk factors. Her childhood was nurtured well, no problems in her personal life, no problems in her professional life. But her anxiety was inconsolable. It was untreatable. None of the therapies, conventional pharmacological therapies, psychotherapy, nothing really worked. So actually, Brian Weiss then did something quite unconventional. He started using hypnotherapy on the patient. And it turned out that during the hypnotherapy sessions, this patient would travel back to her past lives. And apparently, while there was no risk factors for psychiatric illnesses in her current life, there were a lot of risk factors in the form of traumas, multiple complex traumas in her past life. Now, well, this is pseudoscience, yeah? This is not real science at this point. But what Brian Weiss started doing was that he started treating the patient, resolving the traumas in her past life, and eventually he came up with the idea that our past lives affect us, and in each life, there is a master. There is an event which controls everything, which then decides what will happen in your next life. And that's why it's titled Many Lives, Many Masters. And there is one sentence from this book you can all read over here. We are not sent from each plane with equal powers. Some of us possess powers greater than others because they have been accrued from other times. Well, I do believe in what's mentioned over here, but I definitely think that Brian Weiss made a huge mistake because it was not many lives, many masters. It was actually many lives and one master. And that is the epigenetic and metabolic imprint which we receive from our parents and even their own parents. And this is what I'll try to convince you over the next 30 minutes or so. I'll start with what my lab works on. So there's a little video which will explain everything. All right, so I think it's probably very clear that we are interested in neuropsychiatric disorders and their susceptibilities, and we believe that it is not only dependent on the risk factors which we acquire, but also the risk factors which are passed on from the previous generation. So if I would put it like, events like trauma, exposures like high-fat diet, they cause changes in our molecular biology in a way that we become more susceptible to neuropsychiatric disorders, but then they also potentially impact our germline in a way that the next generation is also at increased risk of neuropsychiatric disorders. Well, this idea is not novel. I'm not the one who started working on that. This was actually proposed centuries ago by Lamarck, that different kind of exposures, which lead to acquired behaviors in us, can potentially be transmitted to the next generation. However, this was challenged by Darwin, and it didn't really survive. The whole theory, it kind of blanked out. And what else didn't lead to the propagation of this theory is because there was another scientist named Weismann who proposed this Weismann barrier. So one of his discoveries was actually quite promising, because he found out that germ cells, they can proliferate, but at the same time, what he proposed was that the signaling is only in one direction. So germ cells, sperm and oocytes, they carry the information by which other cells are formed, but once the germline and somatic cell communication takes place, it cannot be reversed. Meaning, if there is an exposure, which all of us are having right now, it will not impact our germ cells. It's only one directional. This was called the Weismann barrier. As a result of Darwinism and the Weismann barrier, Lamarckism pretty much died down. And eventually it took another 100 years or so until people started talking about it again. And that was because of the Holocaust. And there were some studies where they found out that in Holocaust survivors, there are certain potentially epigenetic changes, and similar changes are present even in their offspring. However, there was a lot of speculation about that. Can we really inherit trauma? And the speculation was there because the mechanisms were not identified. And that is one of the key themes my lab is working on. So here is a picture from a review which we wrote a few years ago, where we looked at all different kinds of exposures in humans at the parental level, and we tried to estimate if they can lead to heritable effects in the next generation. So it's a review, it's a combination of a lot of different studies, And eventually what we found out was that there is definitely indications, strong indications, that psychological trauma can lead to heritable effects, as well as dietary exposures can lead to heritable effects. Of course, other exposures, like exposure to toxicants, etc., they can also lead to heritable effects. And finally, these exposures do not necessarily have to be negative. Even positive exposures can lead to heritable effects in the next generation, which could be like, which could be advantageous to the next generation. For example, if the father was exercising, the risk of cardiovascular morbidity in the next generation could be lower. There are indications for that in epidemiological studies. Importantly, it remains, at this point in time, I would say inconclusive if the epigenetic mechanisms are involved, if they're implicated in this potentially epigenetic transmission, which is what we are working on. So the important point when we discuss the concept of epigenetic inheritance is that, number one, it involves these epigenetic modifications in the germline. So, in the end, the central dogma that we have DNA, then RNA, and then protein. However, epigenetics means that all these things could be modified without alteration in the genetic sequence, in the DNA code. So through histone modifications and DNA methylation, the way DNA is transcribed can be affected, or at the post-transcription level, there are non-coding RNAs which can prevent the proteins from being formed. So the whole transfer of information from DNA to proteins can be regulated through these epigenetic mechanisms. Now, importantly, in epigenetic inheritance, this is the most important concept, that the environmental insult is in the first generation. It leads to these epigenetic modifications, and then the phenotype is transmitted across several generations. So the environmental insult is only in one generation and not in the next ones, which is what makes it very exciting and particular. Now, I learned all about epigenetic inheritance during my PhD studies when I was at ETH in Switzerland, and we worked on a model of childhood trauma in mice. It is based on impaired maternal care. So in this model, newly born pups, they are separated from their mothers in an unpredictable fashion for 3 hours every day during the postnatal life, like postnatal life from day 1 to day 14. So during the first 2 weeks of life, they're separated from their mothers. So there is the component of unpredictable maternal separation. At the same time, the mouse mothers, they are placed in this cold water, which is an acutely stressful event for them. So the combination of this unpredictable maternal separation and unpredictable maternal stress, which we call as MSUS, it leads to a particular phenotype in these mice, that when they grow to be adults, then they have multitude of different symptomatology, but mainly related to changes in the neuropsychiatric manifestations, in their metabolism, as well as in their immune functions. Importantly, when these mice are mated with mice which were never exposed to trauma, so this is the traumatized mouse, and this is the wild type, the non-traumatized mouse, the resulting offspring also recapitulates all these different manifestations. So I'll briefly go into what those manifestations are. So we know that mice are nocturnal animals. They prefer staying in the dark. However, what we saw that after MSUS, these mice started preferring the brightly lit compartment, which means that they were taking more risks, similar to how humans take more risk when they have borderline personality disorder, Similarly, what we saw over here is that when these mice are placed in cold water, so a regular mouse, unaffected mouse, would try to escape. They try to escape the cold water, and eventually a lot of them actually are able to escape this chamber with water. But the MSUS mice, as well as their offspring, they just remained floating. Their floating time was increased. Something, again, very similar to human conditions, where we see that depressed patients, they don't have motivation, or they kind of give up on things, and this is something similar which we see in mice after MSUS, as well as their offspring. Their social interactions go down, they become isolated, they do not fit into social hierarchies anymore. Their navigational skills and spatial memory go down. And also, their metabolism is altered. What we are showing over here is HDL, and we found out that in MSUS, as well as their offspring, the HDL was altered. Okay, So the question is, fine, you showed MSUS, and you showed that it has a lot of manifestations, which are not only in the affected mice, but also in the next generation. What is causing it? Yeah. So is it possible that these mice who are traumatized, they are bad parents? Or there is actual transmission through the germ cells? So we tried to actually identify that. First, we studied their brain, blood, and sperm, different, the germline as well as different body tissues. And we found out that there were comparable changes in the non-coding RNAs in all these different tissues. Then what we did was that we took the non-coding RNA fraction, we took the RNA fraction from the germline, injected it into the one-cell embryo, which was produced from unaffected mice. Yeah, so this is something similar to in vitro fertilization. So we're taking the epigenetic material from here, injecting it into the one-cell embryo formed by unaffected mice, and this recapitulated the phenotype, meaning this is germline-based transmission, and it is through RNA changes in the germ cells. But what is causing the trauma to induce those changes in the germ cell? That was the next question. In our next study, what we did was that we thought about what are the possible mechanisms of this transmission, of this induction in the germline changes. And of course, like circulation is the biggest connection between the brain, where you perceive the trauma, and the germline. So we did something similar to blood transfusion. So we took the serum from the affected mice, injected it into the unaffected mice, and it led to transmission of trauma, as well as it led to transmission of trauma in the next generation and induced the germline changes. It opened up two more questions. Is there a mechanistic hub responsible for induction of germline changes? Okay, because in blood there are a lot of factors. There are cytokines, there are hormones, like, there are non-coding RNA. So what is actually causing it? The other question, what is the relevance of all this for humans? Yeah. So my background is also medicine. I did a PhD afterwards, but initially I trained in neuropsychiatry. So for me, this was a very important and pertinent question. Now, there are certain challenges when you try to translate studies done on mice to humans, especially a study like ours. The challenges are, does the trauma in mice recapitulate the complex trauma in human conditions? What about the body fluid-based mechanisms? What if it is not the germline, but actually the vaginal fluid in which the effects of trauma are transmitted? Or what if the effects of trauma are transmitted through lactation? Like, when the mothers feed the babies, are they transmitting trauma at that moment? What about biological versus psychosocial transmission? Okay, we tried to adjust for some of that in our mice, like, by doing these RNA injections from the sperm into the one-cell embryo. But can we detect that also in humans? What about confounding influences? In humans, like, trauma doesn't come in an isolated fashion like in laboratory mice, right? Like, people who are traumatized, unfortunately, they also are more likely to become addicts. They're also more likely to have, like, unhealthy lifestyles later on. So what about all those confounding influences? Finally, what about genetic heterogeneity? When we study something in the laboratory mice, they're all genetically similar. Humans are not, right? We have, like, ethnicities, we have all these different influences. So can we find a way in which we can have a human population which is genetically homogeneous, similar to lab mice? Finally, of course, the germline assessments in humans are not so easy. I mean, like, you would have to find people who want to donate, like, their sperm samples for research. Now, I know that their sperm donation is quite common, like, for other reasons, but that is usually paid, and in research we don't do that. So these are all the different challenges. And finally, what about the mechanistic and causal underpinnings? So we tried to navigate, we tried to circumvent across all these challenges by a very multidisciplinary approach. So we have our mouse model, we have 3 different human cohorts, which we are studying now, and we are working on very innovative models like brain organoids, where we want to mechanistically assess which pathways are implicated. This is the world map, looking quite interesting, not similar to the world maps you usually see, because this is the world map of consanguinity, cousin marriages. So consanguinity leads to genetic homogeneity. It is inbreeding, yeah. So we, in the end, decided to study 3 different levels of genetic homogeneity. So we identified a cohort in Pakistan where the genetic homogeneity is highest in the world. So you can just say that people in Pakistan are all distant cousins of each other, more or less. And we collaborated with the SOS Children's Village, where we identified a cohort of children in which the fathers, they passed away within the preceding year, and the mothers, they were not financially viable, so they had to give up their children up for adoption. So it is very similar to the mouse model. There was the component of maternal separation, which was forced, as well as the component of maternal stress, because these women lost their husbands, so they were undergoing this period of grief and stress. And then we identified another cohort of adult men who had, during their childhood, experienced simple or complex childhood trauma. Our other cohort is in Bosnia, where the level of consanguinity is not as high as Pakistan, but somewhere a bit lower, like at the medium level. And there we are studying the offspring as well as the survivors of the Srebrenica massacre. So Srebrenica genocide occurred in 1995. I would say one of the most tragic events of human history, in which 8,000 people were killed in a single night. A lot of adult men especially were killed in front of their children. Some of these children are alive now. Actually, like, the children were spared during that time, but they underwent something quite traumatic. And they themselves have children now. So through collaboration with an organization and a lot of different ethical approvals, now we are assessing families in Bosnia who underwent this Srebrenica massacre. And finally, our third cohort is in Poland, in Wrocław, where we have mother-child dyads. So again, these women, they underwent childhood trauma, mostly in the form of childhood abuse. Again, a very unfortunate cohort, but they are very resilient, and they volunteered to be part of our study. And there we also are assessing human milk samples. I'll share some of the results. So these are the results from the PLMS children, so the children who had paternal loss and maternal separation. And we are looking at their circulating microRNAs. The project is led by my PhD student. This is Professor Kamenska, one of our collaborators at Nensky. And briefly explaining, this is RNA sequencing data. The redder the data is, the more red the data is, the higher is the expression. The more blue the data is, the lower is the expression. And you would see that there is a complete clustering. A lot of different microRNAs, they are increased in the control group, decreased in the trauma group, and the other way around. When we look at another cohort, now this is the cohort where we are assessing sperm samples of adult men who had childhood trauma. We see that there is broadly an upregulation of all these different microRNAs in the trauma group. Now, how are we accounting for the psychosocial transmission? That's where our Bosnia study is very important. There are a lot of different procedures which we follow over there. I would just briefly walk you through it. So after recruitment of families, they undergo several pre-procedures in which the fathers are told to abstain, from sexual activity. There's overnight fasting because we have a lot of different metabolic parameters. Children are giving standard breakfast. Then on the day of the study, we collect semen from fathers. The sperm is pelleted out. And like the rest of the family, they undergo a lot of different procedures. Fathers are again involved back in those procedures as well, where we assess them at the psychometric level. So there are a lot of different neuropsychiatric assessments. We have social assessments like family functioning scale. Are there problems within the family structure? Are there problems in parenting? What are the symptoms in the children? And we do a very comprehensive blood-based assessment. So this is to make sure that we are able to tell how much of the trauma transmission is psychosocial and how much of the trauma transmission is actually through the germline, because this is what we'll receive from the fathers. Similarly, in the study which we are doing in Wrocław, we are able to tell how much of the trauma is being passed on through lactation, so through body fluids, because we'll be able to compare the non-coding RNAs in the milk samples which we are collecting and compare those with the epigenetic signatures in the mother as well as the children. Right, so doing all these studies in the mouse model and in the humans, it actually has helped us identify also the mechanistic hub. What pathway in the body is responsible for imprinting those changes to the germline? And it turns out that it's most likely the lipids. So here we are showing non-coding RNAs, which we assessed in serum, in sperm, and in milk. Now keep in mind, these are 3 different cohorts. Pakistan, Poland. They are not one cohort. And they are different body fluids, serum, sperm, and milk. But we consistently found out was that there is this microRNA 223, which is altered in a similar fashion across like all the different cohorts. Now this microRNA 223 is involved in biogenesis of cholesterol. So why lipid metabolism could be the mechanistic hub? Number 1, our data tells us that. Number 2, as you would know that our brain is covered by this barrier, right? There is the blood-brain barrier which separates the brain from the blood and other kind of changes in the body. Similarly, we have a blood germline barrier. Our sperm and egg are also separated from the circulation through this blood germline barrier. And what is that one molecule which is able to traverse these barriers? Lipids. So it is not actually surprising that lipid pathway turns out to be like, the pathway which nature chose for effects of parents to be passed on to the offspring. Number 2, when we talked about all these different exposures for which heritable effects are known or have been documented, they can all potentially affect lipid metabolism. Diet, trauma, exercise, smoking, they can all affect lipid metabolism. Finally, in our mouse model, we saw changes in HDL, as well as in our model, like our cohort, the PLMS cohort of children with trauma, we also saw changes in HDL. Now, HDL, we all know it as the good cholesterol, right? LDL is the bad guy. HDL, high-density lipoprotein, is the good cholesterol. However, HDL also has the ability to bind these microRNAs, these non-coding RNAs in the circulation, which we previously showed in our cohorts to be altered. And there have been studies, like very, I would say, scientifically robust studies, where they showed that microRNAs can go from one tissue to the other and alter gene expression in the other tissue. For example, there is a very interesting study, I'm digressing a bit, but the non-alcoholic fatty liver disease, some of the people, like a lot of scientists, actually believe that it is due to microRNA transfer from the adipose tissue to the liver. So it is known that microRNAs can travel between tissues. They are carried by lipids, and we saw changes in the lipids. So the microRNAs are produced in the nucleus. They can be packaged into extracellular vesicles, or they can be carried by HDLs or by apoptotic bodies. Eventually, they go to the recipient cell where they're taken up by this SRB1. All these things are related to the lipid metabolism. And what we are doing in the lab right now, my graduate students, that they are collecting all these different non-coding RNA conduits, non-coding RNA carriers. And we already performed these injection studies. So this is the MSUS model with trauma. And we want to see if injecting these non-coding RNAs would lead to trauma transmission in the next generation. But also, if we would be able to reverse it through exercise or enhance it through high-fat diet. And we have some very interesting results. So here, we are showing the glucose tolerance test from the next generation, from the offspring. Yeah, so what happens in glucose tolerance test is that you give a bolus of glucose, glucose rises. At a certain point, insulin kicks in because of hyperglycemia, brings the glucose levels down. What we see over here in the MSUS mice, the glucose levels are raised abnormally high, they don't go down. In the high-fat diet model, again, they're raised abnormally high, they don't go down. In the exercise model, we see that they're actually not rising. They're not getting abnormal. So meaning now we are at a stage where we are able to identify some of the therapeutic options which we can use to prevent this trauma transmission. What are those therapeutic implications? Like you as future doctors or as current doctors, I think they are most interested in that. So one is, we clearly know that lipids could be, a risk factor for this trauma transmission. We also know that potentially exercise can prevent this trauma transmission. I mean, we don't know whether it can fully prevent it, but at least partially it can lead to some kind of mitigation. We do know that this involves germline, so eventually perhaps we will reach a point where we will be able to edit the germline, through this CRISPR-Cas technology and all these innovative tools. At some point, we might be able to prevent this trauma transmission through germline-based interventions. And eventually, think about the clinical implications. When you go to a clinic, you always take family history, right? You ask patient whether there was family history of diabetes. Do you also take family history of trauma? Perhaps it's time that also as doctors we start looking into these kind of histories, the epigenetic histories or exposure-based histories, because they do dictate the risk of disorders even across multiple generations. Finally, we also made a very interesting discovery. Now this is also based on this trauma cohort, this PLMS cohort. So you see over here that we separated the children according to those who developed depression after trauma versus those who didn't develop depression after trauma, right? So not everyone who gets trauma develops depression in the end. So there are people who are more resilient, and then there are those who are more susceptible. So we did a sequencing of non-coding RNAs, and we identified that there are some microRNAs which are indicative, which are signatures of this resilience after trauma. So potentially, we can imagine a future in which people who undergo traumatic events, they could be given these RNA-based therapies, microRNA-based therapies to enhance resilience. I also want to briefly mention that we are working on these very innovative models like brain organoids, in which you would take a skin fibroblast, reprogram it into induced pluripotent stem cells, and you can grow a mini brain in a petri dish. And this is another model which we are using to study the effects of trauma, and also at which level of development are the changes induced, and eventually if there will be a way to reverse that. Finally, I want to just highlight some of the implications of our studies, our research for society, for humanity at large. I believe several of you would have seen this picture. Yeah, so this is the Afghan girl. She was featured on the cover of National Geographic back in the 80s. She became a symbol of the atrocities, of the traumas children go through whenever there is a war happening anywhere. Several years later, the Afghan girl was rediscovered. By that time, she had a child of her own. But has anyone tried to study this? Has anyone tried to think about the children who are exposed to wars, who eventually become parents, and has anyone tried to look at whether they are transmitting trauma to the next generation? Is this something we need to include in our broad clinical assessments, in our policy making, in things which WHO or United Nations is doing? Perhaps it's time that we start talking about that. At the level of my lab, we definitely tried to talk about it. We actually, when the war started in Ukraine, I initially wrote a very comprehensive review about the neuroscience of trauma and the Russian invasion of Ukraine. It was published in Nature Human Behavior. Eventually, I also published another comment in Nature about the invasion of Ukraine and why is it important to especially support the mental health of children and adolescents, because that's where you can save generations. By the way, this was also covered in a lot of Polish media. All right, so with that, I would come to the very last slide. So a lot of work which I have shown over here, this was performed by my, I would say, tremendous lab. They are a group of extremely competent people. This is how we look on a given day. We work on very serious topics, but we are a happy lab. And of course, there's a list of our collaborators across the world, from US to Australia. I'm very thankful to all these organizations in Pakistan, SOS Globus Children's Village in Austria, and the Survivors of Srebrenica Foundation in Bosnia and Herzegovina. Of course, my funders as well. So what I've decided is, because I was thoroughly impressed by the level of engagement during my workshop from the students over here, that I will stay back after this lecture for as long as needed to discuss, to answer questions. If someone is interested in coming to Warsaw to work in our lab, I'm very open to that as well. Actually, in my workshop yesterday, there was a student who performed quite well. I think the name, if I pronounce it correctly, is Victoria Sibowska. And I offered her a reward that she can participate in this neuroscience conference, which we are organizing in Warsaw. Her registration fee is waived. But of course, the invitation is also for any of you who would like to come to this conference. I'll end up with one last video, and then I'm happy to take any questions from you guys. This is the video of our conference in Warsaw, which is happening on 31st May to 2nd June, and the registration ends tomorrow night. All right, thank you very much.
Thank you very much for this inspiring lecture. You are welcome here. Thank you for the workshop. Thank you very much for spending time with us.
Thank you very much. Yeah. Thank you. Oh, perfect. Very nice. Thank you so much.
Thank you. Pytania? You want questions now or rather after?
I'm available. Like, we can do it informally or however. Yeah, I'm here. So whoever wants to.
Okay, do you have any questions now, or better you want to ask doctor after? Maybe after, because it takes quite a long time now. Okay, thank you very much for the inspiring lecture. And it was the last part of our Congress. After 3 days, 24 scientific sessions, 304 submitted abstracts, and 269 qualified for the sessions, from which nearly 250 were presented on the SIMC, and the SIMC is coming to the end. Thank you for your work, for your determination, for coming to the International Medical Congress of Silesia, and thank you that the student in our Congress can take the title of one of the biggest medical student congresses in Poland. No doubt that students and doctoral students, with the support of our tutors, create science. The slogans of the 69th edition of our Congress was reaching the top. We hope that after attending our scientific sessions on such a broad range of topics, all participants were able to dive into new areas of science, medical knowledge. Thank you very much, and hopefully see you on the 70th anniversary edition of the International Medical Congress of Silesia, SIMC 224. I also want to thank very much my board of Student Scientific Association, board of students Scientific of Doctoral Students Self-Government, Polish Association of Dental Students, all volunteers, all of you engaged in organizing SIMC. Without you, this Congress cannot take place. So thank you very much for your working for 1,000% for last 2 weeks and all the year to do this Congress now. Thank you very much. And now, after Congress, we invite you to the stage for a group photo. And we also thank very much our IT, Michał Nowak and the rest of ITs, who all these 3 days work very hard to doing this Congress available online, which was a very hard task. They had like 15 computers in the same time using. So thank you very much for this very hard and not seen by the public work. Thank you very much. And we also invite you, Michał, to the stage.
Everyone is invited to take a photo. Okay, so all the participants, can you just stay here? Okay, so every part, don't go upstairs. No, you're coming here on the stage right now. Everyone. On the stage, all the participants are welcome on the stage. And I'm watching you guys.
Dobra, to my stoimy.
My na dół.
Czy ja mogę pod Katowice?
Chodź, Karol.
My sobie usiądziemy.
Będziesz
Wiem, ale sam od Petrusia.
Będziesz miała jednak lepszy
Dobra, to ja sobie stanę z boku.
Siadamy?
Tak? Bardziej bebek widać.
All the participants, all the sitting participants as well. Okay, please come, everyone.
Don't be shy. Don't be shy.
You there, you're coming here. Yes, you're coming there. Or you'll be getting a photo of each one, like solo. It doesn't matter.
We want to.
Thank you very much.